Myself Dipankar Majumdar. Welcome to my channel Microbiologist's Forum. Today I will discuss regarding the community sense in bacteria, that is the quorum sensing. We the human beings and other higher animals have the community sense, but what is about bacteria? Do bacteria have any community sense? Before 1970, it was believed that bacteria is nothing but a bag of cytoplasm. In 1972, scientists Kenneth H. Nelson and John W. Hastings of Harvard University first reported about the concentration-dependent bacterial behavior on the production of bioluminescence in a marine bacterium, Photobacterium fishery. Now, what is quorum? We know that in a society like us, to make a decision fruitful, a minimum number of members should be present, and this is variable from society to society. Same phenomenon is applied to the bacteria. According to the Oxford Advanced Learners Dictionary, the quorum is the fixed minimum number of members that must be present to make the proceedings of an assembly or society valid. In case of bacteria, for some properties like bioluminescence, exopolysaccharide production, conjugation, competence, etc., a minimum number of individuals are needed. On attaining a certain concentration, bacteria show many properties. Bioluminescence was first reported by Kenneth H. Nelson and John W. Hastings of Cambridge University in 1970. Bioluminescence in Photobacterium fishery, then it was renamed as Vibrio fishery and further renamed as Ali Vibrio fishery in 2007. Now, the microbial community show the concentration dependent behavior like motility, sporulation, virulence, competence, biofilm formation, conjugation, symbiosis, antibiotic productions, etc. Now, kinds of quorum sensing. There are mainly two types of quorum sensing. These are species-specific quorum sensing and interspecific quorum sensing. In case of species-specific quorum sensing, the gram-negative bacterium that is mediated by AHLs. AHL is nothing but acyl homoserine lactone, and gram-positive bacteria through small peptides. And in case of gram-positive bacteria or interspecific quorum sensing, that is the autoinducer 2 or AI2 molecules. Its chemical name is furanosyl borate diester, and AI2 is global quorum sensing molecule because it is found in both gram-positive as well as gram-negative bacteria. And AI2 was first reported in Vibrio harvey by Basler et al. These are the key players in quorum sensing network. First one is autoinducers. There is a list of autoinducer: AHL, AI2, cyclic dipeptides, bradioxetine, AI3, DSF, etc. And autoinducer synthesis: Lux I, Lux M, ANS, HDTs, AI2 synthesis, etc. And quorum sensing regulators: Lux R, Lux PQ, and negative regulation of quorum sensing by anti-activator protein, etc. Acyl homoserine lactone was first discovered at Cornell University, USA. AHL in gram-negative bacteria are of two types: short acyl chain and long acyl chain. In both the cases, the homoserine lactone structure is conserved, and the carbon atoms varies from 4 to 8 in acyl moiety in short acyl chain, but in long acyl chain it is 10 to 18 carbon atoms in the acyl moiety. And here N acyl HSL, that is homoserine lactone, is shown. Ali Vibrio fishery, it is a free-living facultative symbiont of photophores of Hawaiian bobtail squid, that is Euprimna scolops. And the classification of Ali Vibrio fishery according to 2007's classification, that is domain bacteria, phylum Proteobacteria, class Gramma Proteobacteria, and family Vibrionaceae. And this bacterium is gram-negative, rod-shaped, oxidase positive, and motile by means of polar flagellum, and found globally in marine environments. Now, how the quorum sensing works, and this is the Lux Operon, and Lux Operon is a 9.8 kb fragment. It produces the luciferase enzyme that is responsible for the production of bioluminescence. Lux I, that is AHL synthesizes AHL molecules. The more the bacteria present in the environment, the more will be the amount of AHL molecules. If the bacterium is lesser in number, the quantity of AHL molecules will be lesser. And when the bacteria attain a certain concentration, the concentration of AHL molecules will be sufficient and it diffuses through the cell membrane to the environment. And after that, when the concentration is high, then it returns back to the cell and binds to Lux Operon response regulator that is synthesized by Lux R. This is a protein, and Lux R is a protein that is having two parts. One is the C terminal end, another one is N terminal end, and C terminal end is 1/3 and N terminal end is 2/3. And the AHL molecule will bind onto the 2/3 part of the Lux R, that is response regulator, through helix turn helix motif. After that, it will recruit RNA polymerase, and that Lux R Lux I complex binds on Lux box, and Lux box, it is a 18 to 20 palindromic sequence, and it is situated 40 bp upstream of Lux Operon, and it will bind through helix turn helix motif, and Lux R and Lux I complex then recruits RNA polymerase to transcribe the luciferase gene. After transcribing the luciferase enzyme, bioluminescence is shown. And this is the reaction of production of luciferase, and luciferase produces blue-green light through the oxidation of reduced flavin mononucleotide, that is FMNH2, and a long chain aldehyde by diatomic oxygen. Here the reaction is shown. The total reaction can be summarized as FMNH2 plus O2 plus RCHO, that will produce FMN plus RCOH and H2O, and light. The reduced FNNH is provided by the free gene, also referred to as Lux G. That's all. In the next lecture, I will discuss about the gram-positive bacterial quorum sensing. Thank you. Thanks for your patience watching. Thank you.